All right, I'd like to next bring to the podium Dr. Nicole Berchtold. She received her PhD from the University of California, Irvine with Dr. Carl Kottman and continued on there to do a postdoc as well, and now is a collaborator with Dr. Kottman. Before she did all of that, she actually worked in Switzerland for 2 years to study the neurobiology of learning and memory. And she's now a senior researcher at the Institute for Memory Impairments and Neurological Disorders at UCI, continuing her rich collaboration with Dr. Kottman. She spent almost 2 decades of research focused on how exercise improves brain health and function, and elucidating the molecular changes that underlie these benefits of exercise on the brain. Her recent research has revealed novel concepts in human brain aging, in particular that the brain undergoes marked changes in the 6th and 7th decades of life, and there's also gender differences as well, too. She's the author of over 30 peer-reviewed articles, 5 book chapters in the fields of exercise and brain function and aging. And we're very honored to have her here today. Please give a warm welcome to Dr. Nicole Berchtold.
Do you have to do anything with that?
I think it's ready to go. Okay.
Well, first off, I'd like to thank Sarah for putting together this symposium and for inviting me as a speaker. I'm quite honored to be here. I'm going to be changing directions a bit here, moving from the macro to the micro, and telling you about the biology of the brain and how it changes with aging and with exercise. As Sarah mentioned, I've been studying exercise and the effects of exercise on brain health and cognitive function for about, I'll say 18 years, because 2 decades sounds more. And I think it's interesting to note that this is really a very young field, in that 20 years ago, research on exercise in the context of neuroscience was virtually non-existent. And it was really a kind of a series of studies in the mid to late 1990s that were pivotal for sparking interest in this field. And since that time, this field has just grown enormously, and especially as we learn more about the brain's capacity for plasticity, and as we learn that our lifestyle choices affect the health of our brain. So I'm just going to start with a little bit of background on what happens in the brain with normal aging, and then present some findings from a recent study that we've done where we've looked at lifestyle factors such as physical activity and social and cognitive activity to investigate, can these lifestyle factors reprogram gene expression in the brain? And I'm going to end with kind of the evolving story of a key gene, a key molecule that seems to be orchestrating a lot of the changes in the brain with exercise and seems to be responsible for a lot of the cognitive benefits. All right, so what happens with aging? Drs. Kramer and McCulley have already covered a lot of this, but just to remind you, unfortunately, like it or not, cognitive function declines with age. And there are certain cognitive domains that are more sensitive to decline with aging, and these include formation of new memories and also what we call executive function, which is this kind of umbrella term for being able to multitask, selective attention, being able to disregard irrelevant information, task switching, all the type of stuff that you would really need to do with that martial arts demonstration, changing from one motion to the next, going with the rules changing. Anyway, so that's the umbrella term is executive function for all of those cognitive aspects. And we also know that processing speed and reaction time decrease. Fortunately, there's some cognitive domains that remain relatively preserved. The functional imaging studies have shown us that at a functional level, there's a decrease in the network connectivity in the brain. And this is basically saying that the different brain regions are not communicating with each other as well or properly anymore. And we also know that there's an overall decline in brain health and resilience. So something like reduced blood flow or a stroke is going to have a greater harmful effect in the aged brain than in the young brain. So what are the mechanisms for this? How is this happening? What can we figure out about the biology underlying these changes? Well, we turn to looking at gene expression to try to understand or try to clarify some of the molecular changes that could be responsible for these declines. And for this study, we used human tissue post-mortem, since we couldn't find any volunteers to donate brains while they were still alive. And we compared gene expression in young individuals. Here they've been kind of broadly grouped as 20 to 59, and the aged cases that were 60 to 99. And we used a technology called microarrays, which lets you look at simultaneously the expression of every single gene in your entire genome in all of the cases. So this is kind of what the data looks like. You get this overwhelming pattern of lines going in all directions. You're like, oh my God, how am I supposed to make sense of this? So we started with the most top-down approach, which was to say, let's just look at how many genes are actually changing with age. Is there a lot going on with aging or not really? And we looked in 4 different brain regions because we were interested to know if changes were relatively uniform across the brain or if there were selective changes to certain brain regions. And so the 4, actually I'm only going to really focus on 2 brain regions, and those are the SFG, superior frontal gyrus, which is basically your prefrontal cortex, and that's important for all those executive functions I was talking about. So the SFG, and then the HC, which is the hippocampus, which is critical for encoding new memories. So what did we find? Well, it turns out that there's really a tremendous amount of gene expression change over the course of normal aging. I'm not showing this data here, but the change that you see over the course of normal aging is actually greater than what you see happening in Alzheimer's disease. I mean, we were just blown away by that. One thing that jumps out is that the prefrontal cortex, responsible for the executive function, shows really the greatest number of gene changes with aging. And that coincides nicely with the kind of dramatic change in executive function that you have with aging. The hippocampus, we were just a little bit surprised that there were fewer changes there than in the prefrontal cortex, but it's still quite a bit of change. In green, I've shown the numbers of genes that are showing increased expression with age, and in red, those that are showing declining expression with age. Okay, well, so what does this mean? Is this good or bad? Does this have any relation to what's going on in the brain, going on with cognition? Well, to answer that question, you have to look at what types of genes are showing increased expression or decreased expression with age. What are their functions? So what we found was that genes that are showing declining expression with age were overwhelmingly associated with synaptic function. And this was very true, especially in that prefrontal cortex area. And genes that were showing increased expression were related to immune function and inflammation. And this is bad because declining synaptic function is going to mean your synapses are not working as well. You're not going to be able to encode information as well. All those features that I mentioned that are associated with aging. And increased inflammation is really bad because it puts the brain in a state of reduced resilience and also impairs synaptic function. So it turns out that the gene changes that we're seeing with aging appear to be very relevant to the cognitive changes that we're seeing with aging. So the first question we asked was, sorry, there's a mouse-like thing up here. So the first question we asked was, can lifestyle variables like physical activity and cognitive and social stimulation, counteract these changes, these aging trends in gene expression. So for this, we collaborated with Dr. David Bennett, who's at Rush Medical Center in Chicago, and we used cases from his Memory and Aging project. And this was really a tremendous opportunity because Dr. Bennett has assembled just an amazing collection of cases and brains. Sorry, I always deal with the brains, not with the people. And what's remarkable about them is that these are extremely well defined clinically. So they've been followed longitudinally for 10 to 15 years. So they really know what the course of cognitive function has been with these individuals, what are their habits, their lifestyle variables. And so they actually would come in for an annual assessment for cognitive function, and they used a battery of 20 tests to kind of interrogate all different aspects of cognition to see if there's, or just to see how those are remaining stable or changing. They assessed the extent of social and cognitive stimulation, basically looking at social and cognitive frequency on a weekly basis. I assessed depression, a few other things. And important for us, they have an unbiased assessment of physical activity. So rather than this being a test of an intervention to see how exercise affects the brain, this is looking at kind of the total activity that the people undertake in their day-to-day lives. So it's both exercise activity and non-exercise activity. Basically, non-exercise activity is getting up and walking to the bathroom, going up the stairs, parking your car far away before you go to Trader Joe's. All those little bits of activity that don't get captured in a kind of an exercise, in a pure exercise study. So how did they collect this physical activity data? Basically, they wore this little gizmo on their wrist called an Actical activity monitor, and it just records their activity for 24 hours. This was basically 10 days continuous of monitoring. So we got a really good kind of snapshot of what their exercise or their activity habits were. And then you could see on a year-to-year basis that it's actually pretty stable. People maintain kind of similar levels of activity over time. So for the study design, again, it was a microarray study, so we could really comprehensively look at the gene changes in the brain. And we used tissue from cognitively intact aged controls that were 75 to 99 years. And we looked at the relationship between gene expression and total activity levels, as well as the levels of cognitive and social stimulation that these people had in their lifetime. And we focused on the changes in synaptic and immune genes, since we know that those are very affected during the normal course of aging. And again, we looked at 4 brain regions. I'm going to kind of draw your attention more to the hippocampus and the prefrontal cortex, and less so to the posterior cingulate and the motor association area. Basically, the top 3 regions are important for cognitive function, and that motor association cortex was more of a control region that is not to do with cognition. So for looking at synaptic genes, we basically started with generating a comprehensive list of all those synaptic-related genes that this microarray could tell us about, and we ended up with a list of about 590 probe sets that looked at what 350 genes did. So what do we find? Well, just look at the very first graph, the hippocampus synaptic. We see, well, I'll just go through that graph real fast. On the left, the numbers indicate the number of genes that showed a significant relationship with activity or with physical activity, social activity, or cognitive frequency, and whether their changes were in a positive direction or in a negative direction. And so you can see that all of those variables had an effect on synaptic genes in the brain, but the physical activity was really the variable that jumped out as having the greatest effect. And this was very prominent in the hippocampus. If I pull out and just show you the physical activity effect on synaptic genes across the brain regions, you can see that the hippocampus is just super responsive to physical activity levels. And this meshes very well with what we know from the animal literature and the animal studies, where the hippocampus is kind of the first thing to respond to physical activity, and it's the most studied because it does the most things. It was interesting that the majority of genes across the brain were showing increased expression with activity, with a subset showing decreased activity. And then when we looked at the types of within the synaptic genes, what exactly were the roles of these genes that were changing within the synaptic function as a whole, we saw that there's really a lot of neurotransmitter receptors that are showing upregulation. Also, genes involved in the presynaptic release machinery. This means basically the machinery that releases neurotransmitter into the synaptic cleft and is responsible for conveying the signal to the next neuron. And there were also many genes that are involved in postsynaptic stability and plasticity. So that basically is saying in stabilizing the synaptic connection and making it more responsive to stimulation. So as an example of what the data looks like for individual genes, I'm showing you BDNF and the glutamate receptor AMPA2. On the x-axis, I'm showing you it's basically a scale of activity, total activity, and on the y-axis is showing the relative level of the gene. And you can see that there's a very nice relationship between increasing activity and increasing BDNF and AMPA2 expression. And I picked these two genes because they're particularly important for enhancing plasticity and encoding in the brain. As two examples of genes that are changing that are related to synaptic vesicle trafficking release, here's synapsin 2 and piccolo. Probably doesn't mean anything to you, so I'll go right. But I just thought piccolo was such a fun name for a gene. There's also another one called bassoon, and I think they go through the whole orchestra. But so, kind of putting all this gene expression data into a kind of an overarching model. Here I'm depicting a synapse, a neuron making a connection with a postsynaptic neuron, and we see that the physical activity is really affecting the synapse at all different levels. It is at the presynaptic side affecting the efficacy of neurotransmitter release and how well it's packaged and transported to the synapse, how fast it's released. We're seeing changes in neuromodulators like BDNF and somatostatin, which are very sensitive to decline with aging. And on the receiving side, on the postsynaptic side, we're seeing just a very large number of neurotransmitters that are showing gene changes. Just want to point out the ones in black are upregulation and the ones in red are downregulation. So you can see that really activity is really boosting all the molecules that are associated with synaptic function. So for neurotransmitter receptors, we see just many subtypes of glutamate receptors, GABA receptors, serotonin receptors, dopamine receptors, acetylcholine, kind of across the board. And when you look at all of those genes and their functions, what it's really boiling down to is that, what we think it's boiling down to, is that physical activity is modifying these genes with the end function to build the synaptic machinery, to facilitate plasticity and to enhance synaptic strength. think I'm going to skip that one. So now the important question is exercise and physical activity changing or counteracting the trends that we're seeing with aging? So as I pointed out, in the synaptic genes are showing a decreasing expression with age and kind of a continuum of decline when you go into Alzheimer's disease. On the opposite side, on the right side, the right graph, is physical activity is increasing the activity of those synaptic genes. And it turns out that there's really quite a large number of genes that are targeted by both aging and physical activity. So yes, physical activity is directly counteracting the aging-related changes. And those are some of the gene classes and examples of genes that are specifically affected by both aging and physical activity. So we did the same kind of analysis for immune genes, and we ended up with a gene list of 760 immune and inflammation-related genes to look at. And again, we found that physical activity is resetting immune and inflammation-related genes just as it's doing with the synaptic genes. Now, what's interesting to note are a few things here. This is looking at the number of genes, again, that are showing a change in the different brain regions. The hippocampus, again, is showing the most response to physical activity. And when you look across the brain regions, the majority of these genes are downregulated with activity. And this is really important because the downregulated genes are in these different classes, like the complement and the toll-like receptor signaling, interleukins, MHC class molecules. These are all signaling systems that increase inflammation in the brain and interfere with cognitive function and interfere with brain health. So we're finding that exercise, or just plain physical activity, is counteracting the age-related increases in those systems. And we believe that that is another way that physical activity is maintaining the health of the brain. So just to briefly summarize those findings, we are showing here that physical activity does reprogram the brain at the gene expression level. We're seeing this in the human brain, which no one has been able to do before. We're seeing a specific targeting of synaptic and immune function related genes. You see responses across the brain, but really the hippocampus is showing the most robust response. And the net effects are to strengthen synaptic function and to facilitate plasticity, to rebalance the excitability of those signaling networks, to reduce inflammation, and to counteract the age-related trends. So I'm going to turn now to the literature and what we know from epidemiological studies in humans. And probably going to go pretty briefly over this since it's already been covered in a good amount of detail. But physical activity and cognitive function and cognitive stimulation and social activity, they all kind of work together to build cognitive reserve. That basically means you have more synapses that you can afford to lose before you're starting to have functional problems. So from the human studies, we're seeing that exercise and physical activity is really emerging as a very powerful component, similar to what I was showing you at the gene expression level. And I don't think I'll go over those since Dr. Kramer has already covered that. But I'd like to turn to the animal literature, where we just know a lot more about the molecular and the biology, the molecular effects and the biology of what's happening in the brain with activity and with environmental enrichment. Basically, what I'm talking about with physical activity, social and cognitive stimulation, that's what they refer to in the animal literature as environmental enrichment. So you see really extensive changes in the animal brain with these interventions, that you're seeing increased cortical and hippocampal thickness, you're seeing more spines, which is the site of synaptic communication, synaptic connections. You're seeing increased numbers of neurons, especially in the hippocampus. You're seeing new vasculature. You're seeing facilitated plasticity as measured by long-term potentiation, or LTP. And in the animal literature, you're really seeing very consistently enhanced hippocampal-dependent cognitive function. So in the animal studies, the hippocampus is really is very responsive. A couple of mechanisms that have emerged to be involved in all of these effects of exercise and enrichment include upregulation of neurotrophic factors, including BDNF, insulin-derived growth factor, and VEGF, and upregulation of many neurotransmitter receptors, changes in upregulation of the synaptic vesicle trafficking release, and modifications of signal transduction pathways to enhance signaling. And the exercise is really turning out to be, when you look at environmental enrichment, where you have exercise and social and cognitive stimulation all kind of intermixed, everyone wants to know, well, which is the most important one? You know, can I just get away with being really studious, or do I really have to go out and run? Or can I just socialize? And it turns out that while they all appear to interact and kind of interacts kind of in a synergistic way, exercise really appears to be one of the most essential components of it. And we think what's happening is that exercise is putting into place kind of the biology and for learning and for encoding, and then you need this other stimulation in order to take advantage of that. So exercise is facilitating learning. And there's an emerging story that one molecule, brain-derived neurotrophic factor, BDNF, is really a key molecule that's underlying the benefits of exercise on the hippocampus. And just briefly, BDNF is it's a neurotrophic factor, and it was first studied in the context of it promotes the health and survival of neurons. But then in the late 1990s, it was discovered that BDNF actually has a really critical role in the hippocampus in encoding, in synaptic plasticity. So one of those pivotal studies that I was talking about that sparked interest in the field of exercise and neuroscience was a report in 1996 by Carl Kottmann's lab that physical activity upregulates BDNF expression in the brain, in the hippocampus especially. Another pivotal discovery was here at UCLA by Dr. Gomez-Pinilla, who showed that BDNF induction with exercise is actually a necessary component for exercise to enhance cognitive function. And he did that with infusing blocking antibodies to BDNF, so to prevent BDNF from signaling in the hippocampus. And we've shown this recently with another technique using siRNA to BDNF. So what we think exercise is doing through BDNF is that it's turning on faster, it's increasing BDNF, which is this plasticity molecule. So exercise is essentially turning on the mechanisms for faster and more efficient encoding. And we've had a really good example of this recently in a study where we used, it's a task called subthreshold learning. So basically, it's a paradigm where a sedentary animal cannot learn the task because it just isn't there long enough to get what's going on. So it's subthreshold to encode it into long-term memory. But an animal that has exercised, has had access to a running wheel for 3 weeks, can learn this task. So it's enabling the subthreshold learning. Clearly, the brain is getting more efficient with the information it's getting, and it's just able to put it in there faster. And again, that subthreshold learning is BDNF dependent. So BDNF is now coming up a lot in human studies. because it's all well and nice that BDNF is important in animal studies, but what we really care about is what's our BDNF doing and does it matter? And yes, we're seeing that in the Framingham study that decreased serum BDNF is associated with an increased dementia risk. That, as Dr. Kramer mentioned, that physical activity increases serum BDNF levels. And then in the microarray study that I was discussing earlier, there's a very high correlation of BDNF with physical activity levels. And I like to point out that actually the correlation was very high with cognitive activities as well. So both physical activity and cognitive activity increase levels of BDNF. So just to conclude, physical activity and to a lesser degree cognitive and social enrichment reprogram gene expression in the human brain. The effect is seen across the brain, but the hippocampus is especially responsive, and this is consistent with and expands on the literature from animal studies. And BDNF regulation and availability is really emerging as a really important convergence point for how exercise affects brain health and function. So the bottom line is, unfortunately, stay physically and cognitively active to maintain a healthy brain. And I'd just like to end with acknowledging a few key collaborators: Carl Kottman, David Bennett, Aaron Bookman, Pat Boyle, Dan Gillen, and Ella Prieto, and of course, our funding sources. And I'd like to open up to questions.
Hi, I see there's a significant correlation with BDNF with both physical and cognitive levels. Do you happen to recall the correlation coefficient? What's the size of the correlation coefficient?
Well, you know, in the uncorrected data, it was an R squared of 0.48 or 5. And then when the data is adjusted for age and education and cognitive and social activity, then the correlation is just skyrockets. But I don't remember that one offhand because it's it got dramatically strengthened by correcting for the kind of those conflicting variables.
In other words, you could account for about 50% of the animals, more or less?
Yes.
Okay, good, thank you.
This is a modern day question. Is there a correlation between video games and cognition?
It depends on who you ask. If you ask the kids, for sure. If you ask their parents, they'll say no way. Actually, there have been some interesting studies with video games and cognition, because there's this huge debate on is it destroying our kids' brains or is it kind of innocuous. It turns out that kids that play video games have faster processing, obviously faster reaction times, and they have a bit faster or improved cognitive function, more on like the executive function level. I think that they've been using computer games as kind of an intervention in older adults, and are investigating whether that is going to promote, plasticity, and whether that can transfer to other tasks. So there is some evidence that it is good.
Because you expect that BDNF actually enhances encoding speed and synaptic plasticity, do you think that combining aerobic exercise and systematic cognitive activity, is that likely to lead to additive effects or actually synergistic, more than the additive effects on cognition?
Yeah, it's a really interesting question. I think that it's going to be synergistic, because I think that cognitive stimulation on its own is not going to have as broad of an effect on that whole underlying synaptic mechanism it's not going to have as broad an effect as exercise. So while it will enhance that whole system, I think if you have the exercise component, then it's really just going to boost it. So I think it's probably synergistic, but it's kind of more of a gut feeling. I think we have to parse that out a little bit better still.
Hi, you referred to the Rush Memorial Study, and I was wondering, when participants go in only once a year for an annual, over a long period of time, if they had insomnia the last few nights before, or they were undergoing some emotional stress that week, wouldn't that have a pretty serious impact on testing their memory?
Well, definitely, yeah, definitely very recent events, sleep and stress are going to impact your memory function. But the important thing to know is that these patients or these individuals are looked at every year. And so there might be a blip 1 year of change down. When things normalize in their lives, they blip back up again to kind of a previous level of cognitive function. And also, they're looking longitudinally. They can see what the stability of the cognition is on all these different domains of executive function and memory and about 4 or 5 other domains. So we're really interested more in what's their slope of change or their stability with aging, rather than one specific assessment.
Question. We're beginning to fall, I think most biologists are falling into the trap that plasticity is actually good for you.
That what is good for you?
Plasticity is actually a
Yes.
good thing. And I think one of the things you have to keep in mind is the fact that we can have what's called maladaptive neuroplasticity. We see that tremendously in traumatic brain injury with children. We see it in the prodromal state of many diseases. What would you do in terms of your work or thinking of neuroplasticity to begin to delineate, because everything obviously in the brain is being turned on, what is adaptive in a good sense, what is adaptive in a maladaptive or bad sense? And the fact that, aging is probably like our rats, it's really just a sedentary lifestyle that's been, instigated upon that particular being that's going to be, adapted to in some fashion. So again, the question of how do we delineate adaptive, maladaptive types of neuroplasticity?
Yeah, it's an excellent question, and now I regret having skipped that one slide where we were thinking about that very thing. We're like, well, increasing plasticity is good to a point, but if you have everything just too malleable, this is a problem, and you have problems with seizures, and that is actually something that does occur with normal aging. You have increased propensity for seizure, and it's kind of hyperexcitability of the networks. So we looked specifically to see if some of those gene changes acted to rebalance the excitability of the networks. And so we did find that there's a lot of GABAergic receptors that are also upregulated, and also neuregulins that are, neurexins that are upregulated with the physical activity as well. And what's important about those is that those function to curtail excitability. And so we think that while you're getting increased plasticity, you're also getting kind of more controlled plasticity and curtailed hyperexcitability. But it's a very, very important question. And it's something that's very difficult to study, in the paradigm that we're using here with the end state, just looking at gene expression levels, and really something that has to be looked in animal studies.
Just another synergy question on possibilities between exercising alone or in groups, the social network connection, and how that plays into cognitive function.
Yeah, it's an interesting question. We were actually just arguing about that a few days ago, because I said, well, I like to exercise alone. I really don't like exercising with people. And the other person said, what? No way. You want to exercise with people for the motivation, et cetera. So probably exercising in the context of having, other people around, having some social and cognitive stimulation is probably going to capitalize more on the benefits than the kind of the solitary. But I think it's also going to just depend on the personality and whether being amongst a whole bunch of people is a stressor or relaxing,
I just had a quick question on when you're looking at physical activity with gene expression, how much do you take into account the function of time? So if physical activity for an hour versus 2 hours, and how fast does it decrease in that influence of heightening or?
Well, in the human studies, we were looking at total daily activity, so just a measure of kind of total activity. So it wasn't looking at, distinct hourly or anything activity, but I guess you're probably asking more about from the animal studies. From the animal studies, generally you let the animals have free access to a running wheel for 3 weeks, and then kind of the next day, which can be anywhere from 3 to 12 hours after the exercise, is when we're looking at the changes. But it's interesting is that with respect to BDNF, we wanted to know how long is it sticking around? Is it going to tell us, can we find out something about how frequently we have to exercise? It turns out that when BDNF is upregulated with exercise and then you stop exercising, it stays high for a while. It stays high for it stays high for several days, then it starts to decline, and then after about a week it goes back down to baseline. But now here's the really interesting thing, is that if you exercise again, you don't need nearly as much exercise for BDNF to, boom, shoot right back up again. So there's some kind of a memory for BDNF induction. And so, even if you haven't, it's kind of consoling, because if you have an injury, you're like, oh my God, I'm out of it for 4 weeks. I've just completely screwed my brain. I've lost all the benefits. It's not true. And we're looking to see right now how long is that memory for BDNF induction, and does that depend on how active you were in that first bout of activity? How much is it modulated by the time in between, and how much of the second activity do you need for it to respond?
Hi. So I can't help but want to associate these studies with all the studies on exercise and depression. And so then, and now exercise is looking like it's protective against depression in the future. And then also about how there were the acute studies with ketamine relieving depression, and they thought that was via BDNF being enhanced, upregulated somehow. I forget the details. And so then I'm just wondering, and it's a tangent, but if you'll just bear with me, then does depression just become a disorder of plasticity?
Well, that's kind of a tough question to answer, because depression is extremely complex, and it really can't be boiled down to just one molecule, even though scientists love to boil things down to one molecule. But I do think that at some level, depression is a problem of plasticity and communication between brain regions. And I think it is maybe a problem of too much and maybe improper activity in the frontal areas, and then too little communication with the hippocampus, and kind of overall decreased functionality of the hippocampus. So that's kind of my big view of depression. And the exercise is going to put into place some protective mechanisms for depression, but it doesn't prevent it. Depression is kind of a bigger beast. But it does protect to some degree from stressors and stress-induced depression.
Can I just ask a question while I have this? And I do have to be around. Seeing that you're in Irvine and Dr. Daniel Heyman's clinic is there, do you interact with him at all? Or?
No, I do not. I do not at all. Maybe we could talk about that. Okay. I'm not actually familiar with him.
You're what?
I'm not actually familiar with him. Oh, see, that's the thing. We don't have television. Because I can't stand those commercials. So I'm missing out.
My question is mainly regarding exercise and your immune system. Is there any study looking at the rate of, let's say, cancer in athletic people versus non-athletic?
Yeah, there's a great amount of interest in exercise and immune function. And the immune function in people who are physically fit and active and athletes is actually quite a bit stronger, and you don't see the same extent of age-related changes in the immune system in athletes and active people. I think, though, that if the activity, the exercise, so the athlete is overly active, or just if the activity ends up being a stressor, for example, like after a marathon, a lot of them get sick, and it's because a stressor is going to suppress the immune system. But overall, people who are physically active and fit and athletes have a much healthier immune system. Oh.